Hello, everyone, and welcome to Chapter 3, the topic of which is the cell. In Chapters 1 and 2, we discussed the different types of microorganisms that fall within the purview of microbiology, and we also discussed the tools and techniques that we use to visualize those microorganisms. In Chapter 3, we're going to delve further into the characteristics of those microorganisms, particularly their structural and physical characteristics. As we have this discussion, we're going to separate the discussion into two different parts based upon the two different types of cells that are recognized by biologists. You probably remember from a prerequisite course that you've taken that there are two major classifications of cells in the living world: prokaryotic cells, or ones that lack a nucleus, and eukaryotic cells, are ones that have a nucleus. As we said in Chapter 1, cells of microscopic organisms can either be prokaryotic or eukaryotic depending upon which classification of microorganisms we are talking about. Before we move further, I'm going to briefly summarize the other differences between prokaryotic and eukaryotic cells that you've learned previously. Because the presence versus absence of a nucleus is not the only difference between prokaryotes and eukaryotes. While it is true that in prokaryotes, DNA is located in the cytoplasm, specifically the nucleoid region, and the eukaryotes have their DNA stored in a nucleus, there are many other differences as well. For example, prokaryotes have a singular circular chromosome, whereas eukaryotes have multiple linear chromosomes. Prokaryotes typically have few to no membrane-bound organelles or internal structures, whereas eukaryotes typically have many. In prokaryotes, the cell wall is made of peptidoglycan, whereas in eukaryotes, the cell wall, if it's present, is made of chitin, cellulose, or another material. And prokaryotes divide through a process called binary fission, while eukaryotes divide through a process that you have learned about previously called mitosis. So we are going to start off our discussion with the prokaryotes. And as we talk about the characteristics of prokaryotes, I want to remind you that 2 out of the 6 categories of microbes that we discuss in this class are prokaryotic. That is, the bacteria and the archaea. However, when we have our discussion that follows, the descriptions that we give are going to be mostly focused on bacteria. And the reason for this is because bacteria are, well, first of all, we know far more about bacteria than we do about archaea. They're much more well studied. But also, bacteria are far more important in human disease processes, and so these are going to be our focus. And I'll remind you also that when we talked previously in Chapter 1 about bacteria, we noted that there are 3 main shapes that bacterial cells can have. And those were the coccus, cells that are spherical or ovoid in structure, bacillus, ones that are rod-like, and spiral ones, the ones that are corkscrew shaped. However, shape is not the complete picture of a cell's morphology. There are 2 different components that go into what we call as a cell's morphology. There is the shape of the cell, but there is also the arrangement of the cells and the relationship that the cells have to each other when they are in aggregate form. Shape and arrangement are both important signifiers of identity for different species of bacteria. Shape is often the first clue as to the species of a bacterium, And so in the following slides, we are going to be taking a look not only at the shapes, but the different arrangements that those shapes can have. Out of the three different main shapes, cocci, bacilli, and spiral-shaped bacteria, we're going to begin with the cocci. When we're talking about different arrangements of bacteria, what defines the arrangement is the plane of division and the number of times a cell will divide before it separates from its counterpart cells. So here we have a single coccus bacterium, and two possible arrangements for coccus bacteria are the diplococci arrangement and the streptococci arrangement. Diplococci is what you get when a single cell divides once, splits into two through binary fission, and then those two cells remain stuck together in this arrangement. Diplo means two, and so that's where the name diplococci is derived from. Streptococci is similar to diplococci in that there is a single plane of division, but it involves many more than just a single replication. Streptococci is an arrangement that you get when a coccus bacterium divides multiple times and the cells that result from those divisions remain attached in chains. It is also possible to have more than one plane of division. In the tetrad arrangement of cocci bacteria, there are two planes of division, so that the cell divides once in one direction, and then those same cells divide in a different direction. And all four resulting cells remain adhered to each other in the tetrad arrangement. You can also get a sarsenate arrangement, which is kind of like a double-decker tetrad. Instead of a cluster of 4, it's a cluster of 8 that are stacked on top of each other. So there are two planes of division, one in the horizontal direction and one in the vertical direction. The final arrangement that's possible for cocci bacteria is the staphylococci arrangement. And the staphylococci arrangement doesn't have a defined plane of division. So, in other words, the bacteria divide repeatedly in any old direction and remain clustered together, kind of like a bunch of grapes. So next we have the bacilli, or the rod-shaped bacteria. These bacteria can also have different arrangements, such as the single bacillus, which is the simplest possible arrangement of bacilli. This is where the single bacterium is not related to and not attached to any other bacteria. Diplobacilli, much like Diplococcus, is where the cell divides along a single plane and then those two cells remain attached to each other. It's possible to have single bacillus and diplobacillus present within the same sample and species of bacteria, as you see in this image right here. We've got a single bacillus highlighted up here, and in the very same sample we have a diplobacillus right here. We also have streptobacilli. Again, remember that streptococci was spherical shaped bacteria that were arranged in chains. Streptobacilli, so strepto meaning chain, is rod-shaped bacteria arranged in chains. Coccobacillus is a variation on the bacillus bacterium, which is sort of truncated and more oval-shaped than rod-shaped. And because of this, it's kind of considered to be an in-between shape between the coccus and the bacillus bacterium, and therefore it is called Coccobacillus. Next, we have the spiral-shaped bacteria. There are 3 different subcategories of spiral bacteria, the first of which is called a vibrio. A vibrio is a curved rod, and sometimes this is actually recognized as a bacillus shape, but here we have it recognized as a spiral. Spirillum is a rigid corkscrew, and that can be contrasted to a spirochete, the third and final type of spiral bacteria, which is a longer, thinner, more flexible corkscrew. So now you're going to test your ability to recognize some of these shapes and arrangements with a couple of checkpoints. Here we have a microscopy image of cells with a particular morphology. How would you describe this morphology? Next, we have one more checkpoint along these exact same lines. How would you describe the morphology of these cells? So now that we have completed our discussion on the different possible morphologies of prokaryotic cells, we are going to take a look closely at a prokaryotic cell and the structures that are found on its surface and within it. What we're going to do here is work from this given diagram of a model prokaryotic cell, and we're going to start on the outside of the cell and work our way into the cell. So we're going to start with things that are sort of outside and on the surface, go over their functions, and then gradually we will move downward and inward until we're inside of the cell and look at the structures in there. So the first structure that we want to talk about, which is outside of the cell, is something called the glycocalyx. Glycocalyx is a word that literally means sugar coat. It is a sticky substance that the cell makes and then secretes to its exterior, and then the glycocalyx layer sits on the surface of the cell wall and offers additional protection to the cell. In the image right here, the glycocalyx is actually referred to as the capsule, pointed out right here. And that's because there are two different forms that the glycocalyx can take. The material that the glycocalyx is made of, the sugar coat, can either be well organized and tightly attached to the cell wall underneath, and in which case it is called a capsule. It can also be disorganized and loosely attached to the cell wall underneath, and in this case it is called a slime layer. So both of these are two different types of sugar coats, but we can think of the capsule as being like a sugar cube and the slime layer as being more like corn syrup. That's the difference in their structure. However, something that the capsule and the slime layer have in common with each other is that they both increase the virulence of bacteria that possess them. Not all bacteria have a glycocalyx, but when they do, when it's in the capsule form, it increases the virulence of the bacterium by protecting them from being destroyed by the immune system of the host that they are infecting. Bacteria that lack a capsule are less well protected, and a great example of this is in the species that we call Streptococcus pneumoniae. Streptococcus pneumoniae comes in two different forms. One of them that is encapsulated or possesses a capsule, and another one that lacks a capsule. And it is only the encapsulated form that is able to cause their respiratory infection that we call pneumonia. In the images on the screen, we see the results of a special biochemical test called a Quailon reaction, which can be used to test for the presence of a capsule using antibodies. And on the left-hand side, we can see a positive reaction, which shows the presence of the capsules around these cells, but not around the cells on the right-hand side. But let's not forget about the slime layer. The slime layer is the other form of the glycocalyx, and slime layers also increase the virulence of the bacteria that possess them. And that's because slime layers allow cells to attach to each other and also attach to surfaces that they come in contact with, which facilitates the formation of something called a biofilm, which is a layer of tightly packed cells attached to each other and attached to surfaces, and their arrangement in this manner Both resist the human immune system and resist disinfection by protecting the cells that are located in this biofilm layer. So this too causes them to become more virulent. In this image right here, we can see a great image from an electron microscope taken of a slime layer composed of Staphylococcus aureus cells that are forming a biofilm on an indwelling catheter. So, catheters or other indwelling medical devices are a common site where biofilms can form if they are left in the body for too long and stagnated, which allows a biofilm to build up on their surface. Another great example of a biofilm that might be a little closer to home is what happens to your teeth after you eat a sugary meal and you run your tongue over them and you feel that kind of fuzzy feeling on the surface of your teeth. That is a biofilm that is forming from the microbes that are inside of your mouth as they grow and proliferate in reaction to the sugar that you have just consumed. So now that we've talked about the glycocalyx, we are going to move on to talking about different types of extracellular appendages, or in other words, appendages that are attached to the cell but stick off of the cell and perform some function. For example, the flagellum. Flagella are these long, stringy appendages that allow the cell to move through its environment by rotating, so they rotate like a propeller, and in doing so, they can move the cell toward or away from substances that it either wants to pursue or wants to get away from. There are two major ways that bacterial cells use their flagella to move in their environment. The first one is called chemotaxis, which describes the movement of bacteria either toward or away from a chemical substance. For example, they may want to move toward a substance that they can eat and consume for fuel, or they may want to move away from a substance that is toxic to them. Phototaxis is also possible, and this is the movement of bacteria towards a light source. This is particularly useful for microscopic organisms that are able to perform photosynthesis because this allows them to move towards their primary fuel sources of sunlight. There are several different patterns in which flagella can be arranged on the exterior of bacterial cells. Monotrichous flagella represents a single flagellum that is projected off of either one or the other end of the cell. So the example that we've been looking at has a monotrichous flagellum. Amphitrichous flagella is one or more flagella that are present at opposite ends of a bacterial cell. Lophotrichous flagella is multiple flagella that are projecting from a single end of a bacterial cell. And paratrichous flagella, finally, are flagella that have no particular pattern in which they are attached to the surface of the cell. They're sort of found in a disorganized manner all over the surface. So these are cartoon depictions of the different arrangements. And here we have some actual microscopy images of these different arrangements. So peritrichous is disorganized, monotrichous is singular, lophotrichous is multiple, and amphitrichous is either end. Another type of extracellular appendage that we can find on bacterial cells is fimbriae. Fimbriae, unlike flagella, are very short, thin appendages, and the function that fimbriae perform is kind of similar to a slime layer in that it allows them to adhere to other cells and surfaces and facilitates the formation of biofilms. The fimbriae sort of get tangled up with the fimbriae of other bacterial cells that possess them, and in this way they allow mutual attachment. Next, we have the pilus, the plural of which is pillae. And pillae are similar in structure to fimbriae in that they are shorter and thinner than flagella, but they have a different function. They are used either for twitching motility, which is exactly what it sounds like. It's the movement of the bacteria through the repeated twitching of this extracellular appendage, as well as for conjugation. And conjugation is the exchange of genetic information between two different bacteria. Conjugation is where a bacterium will take its pilus and it will extend it and attach it to another bacterium and use that pilus as a tube through which DNA can be transferred from one bacterium to another. This is a fascinating process that we'll talk more about later, but it is a very important process because it is a way that bacteria are able to exchange genetic information with each other and generate diversity. Diversity in bacterial populations would be lacking in the absence of mechanisms like this, because every bacterium reproduces by cloning itself and making identical copies of itself. The pilus and the process of conjugation allows for bacteria to share genes, and the consequences for humans can actually be quite negative because it has been realized that bacteria are able to conjugate and share genes for damaging traits such as the production of toxins or the ability to become resistant to antibiotics. Both of those are known to be passed between bacteria through conjugation. In this checkpoint here, I'd like you to take a look at this microscopy image and tell me what arrangement of flagella do we see in this bacteria. So now we're going to proceed even further down through the layers, and we're going to take a look at the cell wall. The cell wall is a rigid layer of material located beneath the glycocalyx when a glycocalyx is present, and on top of the cell's plasma membrane. Bacterial cell walls play a critical function in their ability to survive. They maintain the shape of the cell. They protect the very fragile cell membrane that lies underneath. As you can see in this electron microscopy image, we've got the thick cell wall on top, and then underneath it is this lighter colored layer is the delicate plasma membrane. Cell walls also prevent the cell from bursting by taking on too much water. Essentially, they are a barrier that prevents the membrane from overexpanding to the point where it would actually burst if too much water was absorbed by the cell. The cell wall prevents this. They also serve as the anchor points for flagella. As it turns out, the structure of the bacterial cell wall is so important for its viability that it's a very good thing to target and disable if you're looking to kill a bacteria. And so many of our antibiotics actually go after the cell wall. And by destroying the cell wall, they are able to destroy the bacteria. And this leads us into our discussion of the composition of the cell wall and the material that it is made out of, which we have noted before is called peptidoglycan. Peptidoglycan is a very complex material that is made out of a combination of both carbohydrates and proteins. Specifically, disaccharide carbohydrates are cross-linked with polypeptide or very small proteins to form a rigid material that gives strength to the cell wall. There are multiple ways that peptidoglycan can be formulated and incorporated into the cell wall, leading to two major categories of bacterial cells that are recognized based upon their cell structure. The first is the gram-positive cell wall. The gram-positive cell wall you may recognize as being mentioned in our previous lecture when we discussed differential staining and the gram stain. That's what the structure is named for. Gram-positive cell walls contain many layers of peptidoglycan that are stacked on top of each other, signified here by the big purple block on top of the cell membrane. They are therefore very thick and rigid, and they are interspersed within these things called teichoic and lipoteichoic acids, which play a role in regulating cell division and determining the shape of the cell. So here's a closer look at the gram-positive cell wall, and as we can see, it's very thick and located on top of the cell membrane here. The gram-negative cell wall, on the other hand, has a quite different structure. While the cell wall is composed of peptidoglycan, instead of many thick rigid layers of peptidoglycan, it is composed of just one or very few layers of peptidoglycan, making it thinner and more flexible. The wall is also located in a space called the periplasm, which is sandwiched in between two layers of membrane, the inner cell membrane and the outer cell membrane. The outer membrane furthermore contains embedded toxins called lipopolysaccharides that play a role in the virulence of gram-negative bacteria. The thinness of the wall furthermore leads it to lack the teichoic and lipoteichoic acids that we saw in gram-positive cell walls. So here's a closer look at the gram-negative cell wall. And as we can see, there's an outer membrane, a thin layer of peptidoglycan, and then an inner cell membrane. And here's a side-by-side comparison of the gram-positive on the left and gram-negative on the right cell wall. Now, in this checkpoint, I want you to do some brainstorming based upon what you now know about the structure of gram-positive versus gram-negative cell walls. Penicillins are a class of antibiotics that are most effective against gram-positive bacteria. The way that penicillins work is by disrupting the structure of peptidoglycan. Penicillins are not as effective against gram-negative bacteria. Based upon what you know about the structure of these walls, why would that be the case? We also need to take a moment to mention the acid-fast cell wall as well, because we did discuss the acid-fast cell wall back in chapter 2 when we were talking about the acid-fast differential stain for identifying mycobacteria. The acid-fast cell wall is a special variation of cell wall found in only a few species of bacteria, which is characterized by the presence of a layer of waxy mycolic acids that are found on top of the layers of peptidoglycan in the wall. These mycolic acids have a protective effect upon the bacteria, and therefore special antibiotics must be used to combat acid-fast bacteria compared to regular bacteria that do not have this layer. So we're going to continue to move further inward on our model of the bacterium and talk about what lies underneath the cell wall, which is the plasma membrane. The plasma membrane is a selectively permeable barrier that encloses all of the internal structures of the cell. So once we are past the plasma membrane, we will officially be inside of the cell. The plasma membrane is composed of structures called phospholipids that make up the primary unit. If you've seen these before, it was probably in a prerequisite course, but just to briefly review what their structure is, these are a type of lipid that has a special phosphate head that has an affinity for water and fatty acid tails that repel water. And so the cell membrane is made up of two layers of these little guys that kind of look like two little armies of jellyfish that are facing each other, where the water-loving heads are facing outwards towards the liquid outside of the cell, as well as the cytoplasm inside of the cell, and the fatty acid tails are commingling in here. We call this model of the cell membrane the fluid mosaic model because it indicates the flexibility of the cell membrane and the ability of these embedded proteins to move and shift around fluidly through these layers of phospholipids that are facing each other. The main role of the cell membrane is to regulate which molecules move in and out of the cell at what times, and that movement is performed by the proteins that are embedded in it. So now that we've talked about all of the layers that are outside of the cell, we are now moving into the cell where we find the cytoplasm. Cytoplasm is the name for the jelly-like substance that fills up the inside of the cell. It's essentially the soup that everything else is swimming in. And one of the things that we find inside of a cytoplasm in a prokaryotic cell is the nucleoid. The nucleoid is the region that contains the DNA, including both the bacterial chromosomes and the plasmids, which are extra chromosomal genetic elements. These little guys are tiny loops of DNA that can be transferred, for example, through a pilus to a neighboring bacterium during that process we talked about called conjugation. One key takeaway is that there is no barrier here surrounding the DNA in the nucleoid. The nucleoid is a barrierless space. And to prove that to you, here is an actual microscopy image of a bacterial cell where its nucleoid region has been artificially demarcated with this dotted green line. But there is no membrane that surrounds it. The DNA is just free floating in this space inside of the cytoplasm. Something else we might find in prokaryotic cells are structures called inclusions. Inclusions are spaces where excess nutrients may be stored, and this can include glycogen, starch, sulfur, or other minerals. Another feature that we find inside of prokaryotic cells are ribosomes. The role of ribosomes, you may remember, is to synthesize the proteins for the cell. But it's worth mentioning that the structure of the ribosomes in prokaryotic cells is different than the structure of the ribosomes in eukaryotic cells. Because ribosomes play a critical role in bacterial cells by synthesizing all of their proteins, and because their structure differs from that of eukaryotic cells, they are a common target for antibiotics. Because we can design antibiotics that specifically attack the prokaryotic ribosome, but have no effect on the eukaryotic ribosomes that are found in our cells, and thus limit the toxicity of those antibiotics to us. Another feature that can be found in some bacterial cells, but definitely not all, are endospores. Endospores, as we mentioned before, are these extremely robust resting cells that are formed mainly inside of species belonging to the genera Bacillus and Clostridium during times of environmental stress. As I said before, I like to call them the bacterial bomb shelters because they are tiny compartments that the cell synthesizes in order to store its DNA away and later allow that endospore to germinate and give rise to a new lineage of bacterial cells once the stressor has gone away. This image right here, I by no means want you to memorize or know the stages of, but this gives you an idea of how endospore formation takes place. The cell initiates endospore formation by forming a separate compartment inside of the cell wall where it transfers a copy of its DNA. The spore is then coated, and then the bursting of the mother cell releases the spore, which can then go on to germinate into a new cell that is genetically identical to the original cell once environmental conditions improve. Endospores are so robust that they can resist literally thousands of years of poor environmental conditions and still remain viable. Viable endospores have been found and extracted from the skin of Egyptian mummies that are nearly 2,000 years old. And they've also been found in a 9,000-year-old sample of sediment under a lake in Sweden. So that gives you an idea of just how robust endospores are. So that completes our discussion of prokaryotic cells. which is actually going to be more involved than our discussion of eukaryotic cells will end up being. The eukaryotic types of microorganisms that we are now going to move on discussing are the fungi, the protists, and the helminths. Recall that viruses are actually acellular, so they are neither prokaryotic nor eukaryotic in structure. They are not made of cells at all. So when we're talking about eukaryotic cells, we're talking about the fungi, protists, and helminths. So once again, we are going to start exterior to a model eukaryotic cell, and then we are going to move inside gradually. We'll begin by looking at extracellular appendages, which can include flagella or cilia. Like prokaryotes, eukaryotes can possess these long, singular, thick strands called flagella, which are used for movement. But these flagella are structured differently than the prokaryotic flagella, and they also move differently. They move in a whip-like fashion rather than a propeller. Cilia are shorter and more numerous appendages that are found across the surface of a eukaryotic cell, and they too are used for movement. Eukaryotic cells can also have a cell wall. The cell wall, however, is made up of a different material than that which is found in prokaryotes. In algae, the cell wall is made of cellulose. Fungi have a cell wall made of chitin. Protozoa and animal cells that are found in helminths both lack a cell wall. Other noteworthy features that are found inside of the eukaryotic cell include ribosomes, which generate the proteins, but again, their structure is different from those ribosomes that are found in prokaryotic cells. They also have a nucleus, which is a special dedicated membrane-bound compartment that contains the DNA. And they, of course, have a cytoplasm, which in the same fashion in prokaryotic cells is just the jelly that fills up the inside of the cell. That's the role of the cytoplasm here. Eukaryotic cells also have a host of other intracellular structures that are bound by membranes, from the rough endoplasmic reticulum to the smooth endoplasmic reticulum, the Golgi complex, peroxisomes, lysosomes, mitochondria, and for the most part, a detailed discussion of these structures is going to be beyond the scope of what we're doing in this chapter. Instead, we are just going to focus on and highlight two of these intracellular structures, and you'll see why in just a moment. We are focusing on these two. The first one is the mitochondria. The mitochondria are the sites of a process called aerobic cellular respiration that we will be learning about in our subsequent chapter. And this is where the majority of the cell's energy molecule called ATP is produced. Mitochondria are often referred to as the powerhouse of the cell. On the other hand, chloroplasts are similar but different intracellular structures that are found in only some eukaryotic organisms, ones that can perform photosynthesis. Algae and protozoa, not all, but only some protozoa contain chloroplasts. And the role of these organelles is to generate energy by absorbing sunlight. So why do we focus on just these two organelles, mitochondria and chloroplasts? Well, these two organelles are the focus of an important theory that actually unifies our understanding of how eukaryotic cells evolved from prokaryotic cells. And that theory is called the theory of endosymbiosis. Endo, the prefix endo means inside, and symbiosis means mutually beneficial or working together. So what the title of this theory actually means is that a smaller cell was engulfed inside of a larger cell, and they develop a mutually beneficial relationship. It is believed with very strong evidence that mitochondria and chloroplasts originated when a prokaryotic cell that was free-living was engulfed by a eukaryotic cell, and develop a mutually beneficial symbiotic relationship wherein the internal structure of the engulfed prokaryotic cell provided energy for the eukaryotic cell, and the eukaryotic cell provided protection for the prokaryotic cell. And over the course of millennia of evolution, they eventually became so entwined that they now cannot live with each other. There's lots of evidence that supports the theory of endosymbiosis, including the fact that both mitochondria and chloroplasts are about the same size as a free-living prokaryotic cell. Mitochondria and chloroplasts also have their own set of DNA, which is not true of other organelles that can be found inside of eukaryotic cells. This suggests that they came from a free-living organism that once had its own dedicated chromosomes. They also possess ribosomes, which are a feature that are found in prokaryotes, and furthermore, the ribosomes that are found in mitochondria and chloroplasts are more similar to the structure of ribosomes that are found in free-living prokaryotes today. Their membrane structure resembles that of prokaryotic cells, and they are sensitive to antibiotics. So this brings us to the end of the lecture, where we're going to finish off by tackling 2 checkpoints to review the content we've just learned. In this checkpoint, I want you to compare and contrast the extracellular structures that we've learned about in this chapter by telling me what type of organism can have each type and what it's used for. When I say what type of organism, I mean, is it found in prokaryotes, eukaryotes, or both? Do this for Fimbriae, Flagella, Cilia, and Pili. And lastly, you have isolated a gram-positive cell with endospores and no nucleus. Tell me if this is a prokaryotic or eukaryotic cell, and tell me how you know. And when you have finished this last checkpoint, you are finished with Chapter 3.